Yeah, so that is you, Peter, and Adam. So if you have your presentation for the last panel. Okay, that's right, that's okay, thank you. Ok, así que vamos a presentar ahora a Peter Stadler de la Universidad de Leipzig, Germany. Les recuerdo que todo el programa y todas las semblanzas están en la página del C3, para no perder tiempo en eso, y que puedan conocer la trayectoria de Peter. Adelante, Peter. Welcome.
Thank you very much.
You're welcome.
So, good afternoon, everybody. First of all, my heartfelt thanks to the organizers for bringing me here and giving me an excuse to come back to Mexico for a little while. What I want to do this afternoon is to elaborate a little bit about the complexity of regulation in biology. And I will actually, since we all want to have dinner at some point, and there's supposed to be also a panel between that and my talk, I will not speak about all the aspects of biological regulation, but just at regulation at the level of a single cell, so nothing too complex, but we will see it's already complex and complicated enough. So let's see if this works. Yeah, it does. So biological systems are in a way interesting or somewhat different complex systems in the sense that they have a number of peculiarities that they in part share maybe with social systems, but not so much with chemical or in particular complex systems from physics. And one of them is that they operate at usually several rather well-defined timescales. They offer a large portfolio of responses to environmental cues that are hard or impossible to predict, that are very far away from sort of a simple physical interpretation of like what happens to a gas if you just heat it up or freeze it. So this is much more elaborate. And this also means that the formal description of much of what we're interested in biological systems actually does not easily follow the form that we are used to from the physical sciences. So in systems biology, for instance, you can model stuff with differential equations, but usually you don't really want to because you don't get access to the parameters, the many that you need anyway. So we look at rule-based effective theories, effective descriptions in this area rather than sort of as a basic first principle physics description. There is also kind of very much in contrast to chemical and physical systems, there is a very strong dependence on history. What I do today depends on what I did yesterday and not just on the fact that I'm here in Mexico City today. So simple Markovian descriptions that we are very used to from physics and chemistry just usually don't cut it in biological systems. So memory is one of these kind of magical words that comes in very prominently. And as we've already heard in Enrique's talk before lunch, another difference, and this is something that I will elaborate on a little bit in the next half an hour or so, is we have this diversity of parts, and this diversity of parts actually does matter, and it is an important part of why these biological systems actually work. So we heard about gene expression, right? And this is kind of the physicists' simplified view of how gene expression was supposed to work, and this is kind of what we thought largely 25 years ago on how it actually does work. We have genes, which is just bits and pieces of DNA that encode for a protein, and then there is this somewhat complicated process of first transcribing the gene into an mRNA copy with a little bit of basically boring processing called splicing, then that thing is translated and not really much goes on. So what winds up in the protein, so was the story is essentially what was written on the DNA, and that's it. So then we get the protein. Everything that's interesting is done by proteins. In particular, proteins are also transcription factors, so they attach to the DNA and cause the expression, the transcription of specific genes. So we can actually understand all these circuitry by just essentially saying, okay, proteins cause the transcription, cause the expression of other proteins. So at the worst, there is one network centered at proteins, and that's the only thing that I can use this one. Yeah, so let me play Rockstar here. Yeah, seems to be working, yes? Yeah. Okay. But even in 2000, we knew that this is not the whole truth. There is all complicated cascades of proteins binding to other proteins, modifying them by phosphorylation that signal from the boundary of the cell, from the membrane to the actual DNA. You integrate over multiple of these inputs, and you produce output, which is, in the simplest case, just proteins. And then we learned that life is not that simple. There is actually lots of different regulatory layers, some of which I will just briefly browse through, and it's the interaction of these layers that essentially cause every one of these intermediate steps and a bunch more in between actually to be regulated in a lot of detail, and this is really what allows us to build from simple cells depending on very intricate signals from their environment, also things that are as complicated as multicellular organisms, including ourselves and our brains and the functioning of our brains. So what do we do in order to understand this complication? And so one approach, in particular, pioneered by Davidson and Levine and many others around the 2000s in the context of developmental biology, was to write down detailed specific circuit diagrams that essentially will tell you which transcription factor causes the transcription of which other transcription factors and which ones inhibit the transcription, and you get these kind of very, very complicated networks that very much look like a wiring diagram in a computer or in a very complicated electromechanical machine. And so I will get back to this notion of computation in biological systems in the second part of my talk. So another aspect of this wiring diagram or graphical representation of regulatory mechanisms is that we actually have in detail lots of different levels of description depending on what level of detail we want, which parts we actually want to include in these wiring diagrams, how detailed we want to model the interactions, and also where exactly the boundaries of the system are that we want to talk about. So those are all completely legitimate wiring diagrams, essentially for the Lac operon, which was kind of the first fancy regulatory system that was discovered, and all it does really is allow the cell to distinguish between a rich environment where you have glucose, which is a very convenient foodstuff for yeast or for the E. coli bacterium, and use glucose when it's there, and when it's not there, switch to another system that will actually actively try to input lactose through a specific channel into the cell. And basically what you see with all these arrows is essentially all the feedback mechanisms on basically signaling back from if you have a particular type of intermediate, then this switches on or switches off another part of this whole circuitry. So there is a bunch of other ways of trying to represent what's going on there. I don't want to belabor that, so my point here is just, even though we are abstracting these systems already from differential equations and all the details of the sequences and the structures and whatnot of all these molecules, to just basically simple rules of interactions, even there we cannot settle down on one description that fits all our purposes. So an important part in understanding regulation in biology and in general, I guess, is that we need to be able to switch between different levels of description, between different levels of abstraction. Chris kind of hates the term, but you can think of that coarse graining up and down as a way of formalizing emergence, to just drop the complexity science term once in this presentation. So emergent properties you can simply think as a reasonable coarse graining of a lower level description. In this case, it basically just means making more or less complicated graph these representations of your networks and essentially ensure that going from one representation to the other is mathematically, at least roughly, something like a homomorphism. So essentially you preserve structure. So you don't draw on every level of description a completely different graph that has nothing to do with the thing below, but it's a coarse graining or an abstraction of the lower level. So there is a structural preservation in these graphs. And this is basically something that we can use as a guiding principle for trying to get mathematically theories of that. So of course we also did our own version of that in order to get a bit of a formalization of what we mean by regulatory systems, and essentially basically thinking of that as simple maps between inputs and outputs basically being thought of as discrete, discretized or binned variable rather than in general continuous variables or fine-grained measurements of concentrations, even if you could also do that, so the formalism would support that. And then you basically also get to define where is your system's boundary, so then in your system basically kind of knows or has built in what it actually gets as an input and what it produces as an output. And this is essentially how you can plug these maps together, and basically it's just composition of maps in the mathematical sense that then makes up a whole regulatory system. The problem is if you really want to do something with these, then you very quickly wind up in messy things like this. This is just some part of a transcription factor network of E. coli, so kind of the simplest of the simple regulatory systems that you can think of in a way. And that already is something that by just drawing it, essentially you cannot really say anything about it except it's complicated and maybe complex if we like the term, right? But there is lots of things that you can actually do with these graphs. You can identify focal points in this network. You can identify hubs. This is a directed network. You can also get some idea of the propagation of signals in a causal way through this network, because you actually, in this case, you actually know what comes before what else. You can also start identifying a circle, so you can actually see the feedbacks in there and thereby get a way of decomposing this system into different modules that you can then assign to biological functions. So this is basically all doable from these graph representations. What I also want to spend a few minutes on on the next slides is to just go through some collection of other things that happen beyond these nice sort of transcription factor networks that live on a particular type of entities and have essentially a homogeneous type of interactions with each other. So one of the things that we learned only relatively recently, which means in the last decade or decade and a half, is how important alternative splicing actually is in a regulatory context. So for the non-biologists, what happens is if you express a gene in eukaryotes, including ourselves, this gene actually is not written just as one long word on the genome, but it actually comes as a sequence of individual pieces called the exons. And when that is transcribed to an RNA, so first there is just a working copy of the whole thing made, and then everything that's between the exons just gets cut out and the exons get concatenated, and it's a sequence of exons that is translated into a protein. Alternative splicing simply means that you get to pick and choose which exons you actually include. So you can essentially decide, well, I don't really like exon number 3, so I splice from here to here and just toss out exon number 3, and you get a different translation product. And in general, genes in human genes have at least a handful, some of them several hundreds, of splice variants that all produce different gene products. Some of them actually are just there to downregulate the gene after the messenger RNA has been produced in a cell type specific manner. This process on which alternative splicing product you produce, this of course is again regulated by various different things, by other proteins, but also by other RNAs that interact with these nascent messenger RNA, and then produces yet another diversity of gene products. So from the 20,000 genes that we roughly have, we produce something like 100,000 different gene products, and that only refers to the protein coding genes. And there is even more on the non-coding RNA side. So then, also around 2000, we realized that proteins are not everything. There is a bunch of other regulatory molecules, namely RNAs, that just never get translated into proteins, but that actually function at the level of RNAs. The most famous ones are these microRNAs, which are just little pieces of RNA that specifically target usually mRNAs and just cause a down regulation of their translation. So either they stop translation or they cause even the destruction of the messenger RNA. So you have this layer of, as they call it, post-transcriptional regulation, because it regulates after transcription. So this is basically what these microRNAs do. Of course, they also are produced by a complicated biosynthesis pathway that itself is regulated in a very complicated manner. And actually there is, as we now know, about 10 different ways of making them. What they all essentially do is they get integrated into a protein complex with a protein called dicer, and then basically are produced as these small single-stranded RNAs that then cause either translational repression, so the messenger RNA simply is not translated, or they even cause the permanent degradation of the messenger RNAs, or they also serve as ligands for parts of the immune system and interfere with our own immune response, plus, meanwhile, sort of a a litany of another 2,000 different reaction mechanisms and functions that have been found so far. In the other direction, if we just go back to our simple picture of the gene regulation, there is also the DNA, which was thought of as basically just a permanent copy, kind of a tape that just stores the information, doesn't do anything in itself. So whenever something happens in the cell, you first have to make an mRNA copy, and only then something interesting happens. It turns out DNA is actually not that boring. First of all, DNA is very long in our cells, so it cannot just swim around as a double helix, as basically a thread that's 3 billion bases long. It would immediately shear and break into small pieces. So that thing is actually organized at various different levels and wrapped up into very compact structures called the chromosomes, which are these things. And the part that I'm interested here is actually kind of the lowest level of organization called the nucleosomes. So the nucleosomes are built up from proteins called histones. These histones form these nucleosome bodies, and what happens is that the DNA gets wrapped around exactly twice on each of these histones. So you can think of them, so basically they're kind of like these wheels where you wrap up your garden hose kind of thing. So this is basically what they look like, and the important part now is that these proteins have tails, and these tails can be chemically modified. You can write different chemical residues on different positions of these proteins, and that signals that marks that you can write onto these nucleosomes. They actually have an effect on gene expression. They can actually mark a region of the genome as being expressed as silent, as compactified, so that they cannot be expressed without additional action, and so on and so forth. And it turns out, at least in eukaryotes, there is also a very large number of molecules, proteins, that read specific marks on the histone. And this is basically just a menu of these things. So you see the different types, of the most important types, I should say, of histone marks up here. So there is about a dozen different ones that are fairly frequent. And then you have different molecules that read different types of modifications. And so one wonders, why would you need this if all the DNA does is basically lie there and wait to be transcribed? Why would anybody need a system of reading molecules for these histones? And of course, they're not only read, but of course, since the marks are there in a specific manner, there is also molecules that write them specifically. So you can also think of this as a control structure, because what you find is that many of these molecules that contain protein domains that read or modify chromatin, so histone marks, tails on the histones, they actually have more than one of these functionalities in the same molecule, or it's known that they come in nice stable complexes of multiple proteins. So what you often get is a reading and a writing domain on the same molecule, or you get a reading domain with some effector domain that does something else. For instance, you read and you attract a transcription factor. And what you can do then is essentially you can separate this idea of I read a histone mark and I do something in response to the histone mark. You can actually also go and say, okay, so I read this histone mark, I write some other histone mark in my vicinity, and then I wait for something else to happen, right? And for the computer scientists, if any, in the room, this sounds very much like a rewriting system, right? So you have a signal, you have a pattern, you rewrite this pattern into something else, and then you have some rule that says if there is a particular pattern, then something happens, like gene expression starting. So you have also, in addition to the mode of gene expression by all these networks that we talked about, you also have a mode of gene expression that essentially goes by rewriting the marks on the chromatin. To what extent this is actually used in different organisms, we still don't know. So there is some indications that silencing the chromatin is, of course, important. There is evidence that in repair mechanisms that after a break in the DNA, there is actually a complicated rewriting where information actually does get stored and read as an intermediate step. To what extent this chromatin actually really works as a rewriting system and implements kind of a computer, quote unquote, to which I will return soon, we don't know. But at least it's an intriguing feature that you could even have a kind of a computational device that you can recognize as such on the level of chromatin, just as part of the whole concept of gene regulatory methods. A topic that I kind of have to speak about because it was a large part of my research work, or my group's research work over the last 25 years since I moved to Leipzig, is long non-coding RNAs, also known as link RNAs. If you look at the results of deep sequencing data, then you find that there is hundreds of thousands of these transcripts that just usually do not have any coding capacity. Some of them do code for small proteins, but for a large part of them, we don't know these. Many thousands of them are associated with chromatin modifying complexes like this polycomb complex, so they have something to do definitely with chromatin-based regulation, but certainly not all of them. They also can function in cis, which is just biologists' slang for they exert their function where they are produced at the same location on the chromosome or on the DNA, or they can act just somewhere else. And many of them have lots of other interesting functions. So, for instance, they are the regulators of the mRNA, so they can actually specifically downregulate certain microRNAs that were used to downregulate mRNAs, thereby upregulating a specific portfolio of mRNAs. They work kind of as scaffolding RNAs to bring together protein complexes, in particular some of the chromatin modifying complexes, and so on and so forth. So a whole universe of things where we have very little idea on how they function, but we have very good idea, evidence that they do function. So just one example of these things is this this regulatory RNA unrail that we stumbled across more or less by chance in a classical genetic association study. It just associates with coronary heart disease, which our collaborators in Leipzig were studying in a lot of detail. And if you look at these QTL areas, so mutations in this area have a strong effect on coronary heart disease, but there is no protein coding gene anywhere there. So we started looking at what is there, and it turned out there is this weird transcript unrail, which has very, very many different complicated splicing structures. It makes circular RNA structures on top of the regular linear ones, and it's associated, as we found, with all sorts of diseases. And it actually turns out that Unreal is one of these non-coding RNAs that acts in trans at many different loci by bringing the polycomb complex to this location. Depending on where exactly it binds to the DNA, it can up or down regulate genes to make life a little bit more complicated. The most annoying thing is for a bioinformatician, it actually functions through an enclosed alu repeat motif that's slightly modified. So alus are repetitive elements in the genome that come in tens of thousands or hundreds of thousands of copies, and therefore are the bioinformatician's nightmare because they look the same, more or less, all over the genome. But what that tells us is yet another kind of annoying aspect of gene regulation, and that is all this junk DNA, all these sort of half-degraded, repetitive elements that we just thought of as a graveyard of basically intruders in the genome, they also can have function, and they also can have their crucial part in gene regulation, and they actually do that in a context that actually literally can make us sick. These are actually effect sizes for coronary heart disease risk that are of a factor of 3, 4, so this is like not nothing at all. What makes these long non-coding RNAs so difficult to study is that in general they are very poorly conserved, so it's very hard to find them, it's very hard to trace them through evolution, and they also tend to change their structure, at least in part, over evolutionary timescales. So for a long time this was an open discussion on whether most of them actually do have function. And the standard argument, or the standard route that you go to argue that something has function in biology is to say, okay, so it's conserved over a long time, it must be under stabilizing selection, because otherwise it should have been erased by random mutations that we just accumulate on all the positions where the individual position, the individual nucleotide in the DNA doesn't matter. And so the question was, so if these things are actually conserved, how can we see that they are actually conserved if on the level of individual nucleotides we basically don't see a signal? So what we did is we looked at, well, is there a transcript in a particular region that we can identify as the homologous region in different genomes, just because the delimiting or the neighboring proteins are the same. So we basically know this piece of DNA in human corresponds to this DNA in the bat corresponds to this DNA in the mouse. And then, okay, so there is something transcribed there, but that doesn't really help because the ENCODE project showed us that essentially everything is described. But what you can do is, when you know already which regions you're looking at, you get much more mileage out of the sensitivity of good alignment algorithms, and then you can actually ask, well, is there functional motifs in these species, in these RNAs conserved? And what we found is that you can actually trace splice sites. You can trace this putting together their pieces from the original transcript, and this is what these colored vertical bars are. Every one of those is a splice junction in these particular molecules, and you can trace them down all the way from humans to basically marsupials, and this is chicken here, and some birds, lizard. So basically you can trace them down something like 200 million years of evolution, and doing some statistics over that basically shows that of a subset of 5,000 something well-characterized long non-coding RNAs in human that we basically picked because they had very poor sequence conservation. Something like 2,900 are still there at the root of the mammals. So a very large fraction, at least, of the non-coding RNAs that are expressed in human are evolutionary old, some of them much older than the mammals. So they shouldn't be there, they shouldn't be conserved if they have no function, if this is just noise. So we do have good evidence that these things are functional. The problem is we don't know what their function is, because the function, elucidating their function is at least as hard as doing it for proteins. And for proteins, basically the molecular biology community has worked on that for several decades in literally thousands of labs to go through all the proteins that could be reasonably of interest. So this also works in plants, by the way. So I'm just showing this because this is one of my Mexico collaborations. This was done with Selene Valverde's lab at the Lankebio a few years ago. There is also a connection from all these fancy biomolecules, RNAs, proteins. At the end, we also have to regulate how we digest our lunch, right? And this is, again, an extremely complicated network of chemical reactions that are canalized by proteins that act as the enzymes, right? So these are kind of one of these effectors would be enzymes that just catalyze chemical reactions that act at all these intermediate points here. So essentially everyone, all the blue labels here are enzymes that regulate particular of these chemical reactions. In a very trivial way, the reaction basically doesn't take place if the protein is not there in a reasonable concentration. Plus, of course, you also have lots of feedback where products of a reaction pathway will actually inhibit one of the enzymes at the beginning of this pathway to switch off particular pathways. So there is also lots of feedback that is relevant at the level of small molecules, and that kind of gives us a connection between genuinely biological phenomena like gene expression and the underlying chemistry of metabolism. So the story so far, biological regulation is everywhere. Essentially every process that you can think of is regulated in a very complicated, let's say complex way, with multiple inputs that give you different outputs that switch on or off in particular, say enzymes, transcription factors, signaling molecules in an essentially rule-based way. Biological regulation is distributed over many different classes of substances and therefore many different ways of interacting and implementing these interaction and these feedbacks. And I believe that this actually does matter because this sets different timescales for these interactions and also different persistence. And it also, if you think about in particular the case of chromatin or the case of other chemical modifications, it also implements explicit memory into the system. And basically sort of proves my point that we have this strong history dependence. So this basically, this setup of the system means that sort of the nice statistical mechanics models which have homogeneous parts and essentially homogeneous interactions that are all of the same type and maybe just are drawn from some distribution doesn't really work very well for biological systems. So why should we care about these? Well, one reason of why we should care about this diversity of different regulating mechanisms is that it actually gives us lots of opportunities to design drugs. Nobody says that we only have to address certain types of molecules like GPCRs as the only class of molecules that we're interested in drugging. Right? We have all these interactions in these networks, and essentially any interaction in this network is a potential drug target if we find a way to address it specifically. In order to do this, we have to bring together a global view of the network, because we have to figure out what does it actually mean to interfere with that particular interaction. What's the consequence of that downstream? Does that actually do what we want? Which is basically identifying a drug target, and then we have to find a way of addressing that target, and that means we actually have to also know the specific mechanism and what does that molecule look at in detail. So in that sense, life in the biological systems is a little bit more complicated than just complex system science. We need the complex system science to tell us where to look at, and then we actually need to look with the very classical molecular biology and whatnot methods to actually tell us how we can specifically interfere there. So that's, I think, is an important message, that we cannot take complexity science and use it to replace biology or molecular biology, but it is an important complement to better understand what we're actually doing. So how am I doing on time?
We should close. We do have 15 minutes, and we have to include questions.
Okay, so the rest very quickly. I don't really want to go to the second part in a lot of details anyway. So it's one thought that I think is interesting, and maybe interesting for the remaining discussion without going into a lot of the technical details, which are rather computer sciencey. So living organisms are often thought of as computing. So this statement you can think of as just a metaphor, just says it processes information in some way which is not very productive. So if we want to use this metaphor of computing, then the question is, what is it computing, and how is it actually implementing this computation? The problem there is computer science actually doesn't give us a neat definition of what computing means. It tells us what computing means if I tell it first what computing machine I actually want to use. So if I specify my computer, it will perfectly well tell me any state transition in that system is computation. But if I give you a squirrel, finding out what's computing in there, except for probably its brain, is kind of difficult. A pragmatic way is to say, okay, can we find some processes that we can abstract to rules on small discrete state spaces so that we can interpret as a symbolic computation, as information processing that we can try to understand at the level of rules, like gene on, gene off would basically be such a description. Let me skip these formalities. So a plausible hypothesis would be regulation is actually the manifestation of computation in biology. Unfortunately, this also doesn't help because we don't have a nice abstract theory of regulation either. So this is basically, we have an idea that two things that we don't know are probably equivalent, which is nice but completely useless. But we do have a few subsystems that we can think of as a computer and that we can actually analyze from this point of view. And I just go back to this rewriting system on the chromatin that I mentioned before. So if you think about this, it basically has an architecture of something like a non-deterministic cellular automaton in that rewriting is very local. You just write on the same nucleosome or on the remaining one. One can actually go and estimate what is the storage capacity. So how many bits of information can you actually store in one nucleosome with all the combinations of chemical modifications, and you come out with something like 200 bits. So this is actually a lot for a 200 something nucleotide stretch of DNA. So more or less, in principle, you can write as much information on a single nucleosome that you have in the DNA. You can actually translate this machine into essentially a kind of Turing machine. This was done by Barbara Bryant. If you use this formal model of computation, which now is an abstract machine that basically abstracts a little bit the chemical processes of this rewriting network on chromatin, you can actually rather easily show this is Turing universal. So this would actually be a bona fide computer that can do everything that your laptop or your cell phone can also do. There is a bunch of other systems like this. So for instance, chemical reactions we can think of as a computational device. And this is just some recent work that I did more or less for fun with a couple of colleagues from Vienna and from Odense in Denmark. We were asking ourselves whether building molecules is actually also a computational system that you can analyze in that way, and can you actually show that this is in principle also something that, so building chemical diversity, is this also just a computation that in principle can calculate whatever you want? And the answer is yes. So we look at chemical graph transformation. So we have molecules that via chemical reaction are transformed into other molecules. In chemistry, this is not a random process, but you actually follow specific reaction mechanisms that you can write down as rules. We write them in this way that you have a left-hand side, which is a pattern in a graph that is transformed into a different pattern. So whenever you find this pattern in your graph, like here, you can rewrite that whole graph so that the original pattern is replaced by the new pattern. That's just a formalization of what happens in chemical reactions. What you can do with this, it turns out, you can also implement the Turing machine, so you can also show that this type of rewriting graphs is universal. And then you can ask yourself, well, if I do this not on all graphs, but also only on graphs that are bona fide molecules and only with with chemical reaction mechanisms, can I still at least in principle get something like this nice mechanical Turing machine that can calculate every function? And the answer to that also is yes. So we can prove this can also be done. And this is basically the design of one step of this Turing machine that doesn't matter how that works in detail. So it's not very difficult to actually come up with something that at least in principle works as a series of chemical reactions. So the heterogeneity matters. We have this close connection between regulation and computation, which I think is useful also to guide the way on how to build models and how to think about models to address complex biological systems, namely with models that look much more like computation, that look much more like rule-based systems, that look like programs, rather than modeling them in the classical physics style. And what I think is extremely interesting is this last point here is that biological systems implement computation in different types of hardware. So there's different subsystems, all of which are capable of quite a bit of information processing, and they interact with each other basically making use of the fact that in part they operate at different time scales, at different accuracy and so on and so forth. We don't know to what extent kind of universal computation that's implemented in those systems is actually used, but what we do know is that biological systems compared to our electronic computers are extremely efficient in the information processing that they do by many orders of magnitude. So that sort of as a more science fiction-y last message is that it's actually interesting to ask whether this interleaved use of different computing paradigms is actually something that we could use to make computation also in our devices more efficient and essentially not melt the last glaciers and the the polar caps to do artificial intelligence over the next 20 years. Thanks for your attention. I am not listing all the collaborators that were involved in one way, shape, or form in this work over the last 25 years. I kind of roughly looked this up. This is several hundred people. There was also more than 100 funded research projects, large and small, that contributed to this overall view. So also the funding organizations don't get all their logos on this slide. I just want to single out Lydia, Sonia, and David Krakow from the SFI, who were very much instrumental in this view of biological computation, in particular the work on chromatin. With Christoph and Daniel, I did the work on computational universality, and a lot of that work really was inspired by David Wolpert, who I published basically nothing with, but we talk a lot about these things, and then everybody goes their way when I'm in SFI. So thank you again for your attention, and I'm happy to take questions.
Hola, hola, hola.
Thank you for a very interesting talk. I was wondering, given the fact that most of all possible chemical reactions in the universe will never take place, or all possible configurations of the human genome, let's say we take the Boolean idealization, all possible combinations will be 2 to the 16,000, and we will never explore it, and nature has never explored it. So of course it's attractive to try to identify single genes that will have single effects, but very probably if we move one gene, then it might have multiple effects. So how feasible would it be, the exploration of?
Yeah, so I mean the idea to delete the gene, see what it breaks, is kind of the standard operating procedure in molecular biology. Unfortunately, it also told us that if you use multiple readouts, you also see that most genes have multiple functions, and it depends very much on the assay that you're using, what effect you're really seeing. But this is basically something that's just part of the nature of the beast. So this piling on top of each other of different mechanisms and reusing things in different contexts all the time. This is basically just how evolution works, and this is probably also why biological systems are relatively resilient compared to our engineered systems, which are very monomanic and have everything has exactly one function in there and does one thing. And
but my question is whether you see any inherent limits to the approach of trying to have personalized medicine, or not personalized medicine, but let's say even just to cure some disease when we don't really understand the whole complexity of.
Let's say this is let me answer this at two different timescales. If you ask me with current technology, then there is obviously a lot of limitations because there is lots of things that we cannot efficiently test. I don't think there is inherent limits to devising really personalized therapies. Together with Jens Meiler, we actually in Leipzig, we are working on pipelines that basically say, okay, so let's identify what is broken and then immediately design a drug that addresses the particular gene that is broken. So in this, at least for certain types of cancers, an approach like that actually looks reasonable and also practically doable, modulo, regulatory constraints that you cannot just feed a patient whatever new molecule you dream up with. But if you can find a drug that's already on the market for some other disease, then in sufficiently severe cases you actually can do this, and at least in small numbers of cases where this has been tried, it actually does. These things do work. So there is definitely value in more personalized medicine, in particular for very severe diseases.
Someone else?
Thank you very much for the talk. I wasn't aware of the diversity of regulation of regulating systems in biology, in cell biology. And you mentioned that one of the possible reasons that cells have this diversity of regulatory systems might be due to the time scales on which they act. But how reliable are they in comparison? You mentioned that you can see chemical reactions as a sort of computation, but the fact is that when you do chemistry, you always have side products. You don't always have exactly the product you are aiming for. You always have some deviations, or where you are expressing proteins, you have misfolded proteins. How reliable are these systems in relation to each other?
That's a good question. So we do know that there is side products for almost everything, but there is also rather fancy surveillance mechanism for everything that where there is a danger that broken remnants might accumulate. So you have a specific system for the surveillance of the functionality of tRNAs, because these would be very bad in translation. So they actually get checked at a very, very fast rate. You have lots of specific degradation pathways that recognize unfinished proteins, that recognize proteins that are chemically marked in specific ways by specific processes. There is nonsense-mediated decay that removes sometimes on purpose. So sometimes RNA premature stops in RNAs. So there's a large number of mechanisms that basically are just there to remove garbage all the time. Again, all these things are regulated, so there's actually lots of checkpoints in these systems that basically check where is this really a molecule with a particular type of damage, then it goes into this destruction pathway. So from that point of view, these things are reliable. So we don't have it, for at least as far as we know, there is not a lot of chemical reactions in our metabolism that are not specifically catalyzed by enzymes. And these enzymes are actually reasonably specific, at least in the reaction that they catalyze. They are not always super specific in terms of what are their substrates, and then they will just basically catalyze the analogous reaction for a bunch of different substrates, which probably is, in secondary metabolism, is a desired feature rather than a bug. In the core metabolism, these things seem to be very specific, at least where we have measurements that the substrate specificity is very good. This also goes, for instance, in bacteria have lots of riboswitches which sense particular metabolites or environmental molecules, and they are also very, very specific. So you can find RNAs that they are artificial molecules, but they are very close to the real thing, that actually can distinguish theine and caffeine. Or theobromine. So these basically isomers of caffeine are nicely distinguished.
Thank you very much.
Sorry, here
Peter at the back, very back.
Yeah.
Sorry.
Yeah, I'm just half blind, so, yeah, here you are. Hi.
So I really like the computational view, and I often are accused of taking it too far, I guess, in some scenarios. I wanted to think a bit more abstractly about not just the computational machinery, to know that we have at least one, you mentioned a couple of implementations of Turing machines in biological substrates. But even more abstractly about the programs that can be running there. So if I think about a cellule, it needs to interact with its environment, and all of the regulatory networks that you talked about are meant to be programs that allow the cell to interact with its environment. So do you have a sense of what types of models the cellule is building of its environment, and what type of programs it may be?
I mean, if you think about the architecture of these gene regulatory programs, what this would suggest is that you often have this kind of if-then rules, right? So you don't sense everything in your environment. You have a rather limited amount of kind of sensory inputs, at least if you are a single-celled organism, right? There is some chemical sensing, maybe you see light or something like that. And over evolutionary timescale, you actually built, so evolution built for you a model of your environment, what it means that there is light, that there is this specific chemical that you sense, basically by Darwinian evolution, right? So if your model doesn't predict well how you should behave, you're just dead and get eaten, and that's it, right? So over a few billion years, the models that are fairly accurate representations of your environment, in the sense that you get a very good predictor for this combination of signals means I better do this. That you have internalized. What I don't know is how you get an explicit representation of what is actually built into the cell out of this architecture, so that you could actually say, okay, so what of the environment is this thing really modeling? No idea.
Because the problem itself you described is exactly the same problem that the brain has. It has a limited, very small channel of communication with the world itself, and it is able to. So I guess my question was, how far have we traveled in order to interpret the type of internal representation?
Not very far. If you are an optimist, you would think that explainable eye techniques might eventually help in that direction.
So thank you very much, Peter.